So, welcome back. Yet another topic review video. This is on Unit 6, Growth, Maintenance, and Repair. So the first bullet point I want to hit for this unit is the first one listed, which reads, use models to classify eukaryotic chromosomes and identify key structural features. And so first, I'd like to just point out that eukaryotic organisms are any organisms that have membrane-bound organelles. And so when you are looking at the chromosomes of a eukaryotic organism, they are going to be found inside of the nucleus for 95% of the cell's existence. Right? So that is one of the defining features. They're typically held within a nucleus. The other defining feature of a eukaryotic chromosome is the fact that it is linear. Right? Basically, it's just a long tube-looking thing. Kind of looks like a bent hot dog. Most chromosomes do. Many eukaryotic organisms are diploid, meaning they have two copies of each chromosome that they have. Right? This comes from the fact that most organisms, or rather, most eukaryotic organisms, reproduce sexually, right? Or at least have the option to reproduce sexually. And so they have a mother and a father. And each of those contributes one copy of the chromosome to that organism. Okay? I think that's it. I mean, I guess you should also know the parts of the chromosome. You have genes, which are special regions of the chromosome that contain coding areas that actually code for a protein. And the rest of it is just full of junk. That's important for structure. Right? It's like filler. You need filler between genes. But yeah. Cool. Next point. So our next point reads, create, analyze, and complete models and descriptions of the cell cycling, including interphase, growth and DNA replication, as well as cell division, mitosis, and cytokinesis. And so this model that I have right here is a model of the overall cell cycle. Right? This is basically the life cycle of a cell. A cell is born when M phase is finished. It moves into G0 phase. Now, G0 is the phase where cells exist to do their job. They are the right size. They have the right amount of organelles. They are doing their cell work while they are in G0. When it comes time for the cell to be replicated, as all cells must at some point, not all cells, most cells must at some point, the cell is moved out of G0 and into G1, which is growth phase 1 or gap phase 1. I much prefer growth phase 1 because during this phase, the cell is growing. It adds cytoplasm, it adds some mechanisms that are needed to complete cell division, and it gets bigger. Once it is aptly big enough, it is then moved into S phase, which stands for synthesis phase. And it's the synthesis of DNA. This is where DNA replication occurs. And so in M phase, I'm about to try to make multiple copies of this cell, so I need multiple copies of this DNA. Namely, I'm going to make 2 copies of this DNA during S phase. Okay, so S phase is basically DNA replication. Once I have a duplicated set of the DNA, I have 2 copies of the DNA, the cell is moved into G2, which is a second growth phase. Okay? Has a little more cytoplasm, but mostly here it's building up a bunch of copies of its organelles, and it's making the centrosomes that are needed to move the chromosomes around in M phase. And M phase is either mitosis or meiosis. For the sake of this unit, it's definitely mitosis, which is cell division. The white letters in this model are collectively known as interphase, and the orange letter is cell division. So in S phase, the process of DNA replication is occurring. I have a very rough, simple, clean, dirty model of DNA replication here. Yeah. And so here is our model of our initial DNA with our sequences in it. We have our AT-CG pairings, as you should be familiar with. And this is my original DNA strand, pre-replication. So an enzyme called helicase will come along and it will separate the pairs. And so the strand's broken open. A secondary enzyme called DNA polymerase comes into that gap where they are separated, and it reads the open nucleotides. When it reads the nucleotides, it finds its pairing, and it builds a new DNA strand alongside it. Okay. So the DNA polymerase came in here, went vroom, okay, and it built these two new strands. Okay. So when it read A, it put on this T. When it read C, it put on this G. When it read G, it put on this C. When it read A, it put on this T. And when it read T, it put on this A. And in the other direction, it did the same thing here. Okay. So these inside strands in this model are completely new. They were just built. They're called nascent strands. The original strands, which are still left over, they're still here, are called template strands. Okay. And so this process creates two copies of our original DNA, and each copy is half new and half old. The term for this is semi-conservative. We saved part of it and we made a new part. Okay, that's DNA replication. I started with one copy of the DNA, I ended with two. This happened during S phase. Okay. So that was right here. Okay, so this was my original cell. I had one copy of each chromosome. I did S phase, boom. I duplicated the chromosomes. Okay, so these little X's are two copies shoved together at the centrosome. Yeah? Centromere, yes. So now I have this cell that's going to move into M phase, which is mitosis. And ultimately, the whole point of mitosis is to neatly separate the duplicated copies of the DNA into two daughter cells. And so the process of mitosis has 4 steps: prophase, metaphase, anaphase, and telophase. They separate these two chromosomes that are duplicated in a neat, orderly way. And then it's wrapped up with a process called cytokinesis, which rips the cell in two. Okay? And so my two daughter cells, the two cells that come out of this, are identical to the cell that we started with. So if this was a kidney cell, these are two kidney cells. Okay? The function of this is for growth and repair. When I was a tiny little baby, I didn't have nearly as many cells as I have now. That's how I got bigger. I added more cells, right? And so I had to make new cells. So we just copy it. We take one cell, we make it 2 cells. Those 2 cells then can turn into 4 cells. 4 cells can turn into 8 cells. It's a very quick process. Now that I'm a full-grown adult and I'm not growing anymore, right, I still need to do mitosis to repair the cells that get damaged in my body, whether it be through injury or just through the general wear and tear of life. You know, those cells need to be replaced, and so healthy cells around them will do mitosis and fill in the gaps where my cells are missing. Yeah, and that's it. Bye!